Hi guys, it's M by Jem here, and this is my third video on the second chapter of A-level biology, which is photosynthesis. Now we all learned that photosynthesis is the process by which plants make their food and all that stuff, right? But now we're going to learn in detail how photosynthesis actually occurs. Before that, we got to learn the structure of a chloroplast, and after that, we're going to learn some light-dependent reactions and light-independent reactions. Photosynthesis always takes place in two stages. One is light-dependent, one other one is dark reaction or independent, light-independent stage. Okay. And the last thing we're going to learn is photophosphorylation, which is also in two stages, which is cyclic and non-cyclic. So let's get into the structure of a chloroplast. Um, yeah. So now you guys can see this is a structure of a chloroplast. No, so it's always a double membranal structure. Here you guys can see this is the inner membrane. The third one is the outer membrane. This one, the dark line here. And between these membranes is a intermembranal space. Number four, this is stroma. Stroma is the liquid which is present inside the hollow structure of a chloroplast. You guys can say that, like for example, in cells there's cytoplasm. So in chloroplast there is stroma, and just like the cytoplasm, there's a lot of water and a lot of other organic molecules inside the stroma. Now this one, number five, you guys can see this is the thylakoid membrane. This single one is the thylakoid membrane, the single disc. These stacks of discs which are joined together, these are called the grana. Well, singular is always granum, and the plural is always grana. Number six, which one is shown here? This is also a thylakoid membrane, but it's all just a link, which is like from one granum to another granum, and this link is called lamella. So now we learned the structure that this is the number one, this is the inner membrane. Number two, intermembranal space. Number three, outer membrane. Number four, the stroma, all the cytoplasm of the chloroplast, you guys can say. And number five, which is the thylakoid. Number six, you guys can say the lamella. And granum is the stack of the thylakoid membrane. Remember that granum is the stack of thylakoid membrane. And these granum, why are these granum present? These granum are present so that more of the membrane can fit inside. You guys know that if a structure is coiled, it can fit more, like it can fit with a greater surface area in the same volume in which uncoiled structure is not gonna fit, right? So the main reason of packing something, the main reason of stacking something, is that it can fit into that space. So yeah. Now we're gonna learn that inside the chloroplast, we know that there are a few stuff that were not mentioned in the previous diagram. You guys can see there's a chloroplast DNA, there's a ribosome, and there's a starch granule. Now these three structures are new here. Now what is the reason of a chloroplast? The chloroplast DNA. Now why is the chloroplast DNA present here? The chloroplast, the DNA is also present in the plant cell, right? In the nucleus of a plant cell, and chloroplast is also controlled by the nucleus of the plant cell. But why is there excess DNA here? Well, the answer to that is pretty simple, because this DNA here produces proteins that the nucleus cannot code. So this DNA is also present for producing excess proteins, which the nucleus does not have control over. So the ribosomes here are present because when the DNA present here, when these loops of DNA which are present inside the chloroplast, these loops of DNA send signals for the protein, right? For the production of the protein, the protein synthesis takes place at the ribosome. So yeah, as you guys learned before, that ribosome is is the site of protein synthesis. So this protein is made in the ribosome. So you guys can say that this DNA is present for the production of protein, and then when it sends signals to the ribosome, the synthesis of the protein occurs at the ribosome. The starch granules are present because when the products of photosynthesis take place, I mean when the photosynthesis takes place, the products are formed, and the one of the major product is starch, which is basically stored glucose. So these starch granules form here to store themselves, right? Okay. And these ribosomes, always remember, they are 70s ribosomes. We learned this before too, but yeah. As a reminder, these ribosomes are 70s ribosomes. Now here you guys can see the thylakoid membrane. Inside the thylakoid membrane, remember that there are two photosystems, photosystem 1 and photosystem 2. The photosystem 1, which is right here, is the main photosystem, which was discovered first. That's why it's called the photosystem 1. And the photosystem 2 here was discovered later on, but it comes before the photosystem 1, so it's just named photosystem 2. I mean, of course, if you discover something first, it's going to be named 1 and then the second one. Now, why are they called P700 and P680? Well, this is because the photosystem 1, it absorbs light of 700 nanometers, and the photosystem 2, it absorbs light of 680 nanometers. So yeah, inside these photosystems, I'm just telling you guys that there are two pigments. One is the primary pigment, which absorbs mostly sunlight and most of the light, and the accessory pigments, which help in the absorption of light, which help the primary pigment of absorption of light. Basically, what happens is that when these accessory pigments they absorb light, they transfer it to the primary pigment. So the main source here is the primary pigment. So yeah, okay. Now we're gonna go into the two stages of photosynthesis. I told you guys that photosynthesis, of course, I mean, it has two stages, the light independent and the light dependent. Light independent is also called dark reaction, and the light dependent is sometimes called light reaction. Okay, so we're gonna move on, and here comes the structure of a chloroplast. And what is happening inside here is basically photosynthesis. This is the all of photosynthesis here. It goes from all the way from the light dependent reaction to light independent reaction. Remember that these reactions are always continuous. I mean, uh, without one, the other one cannot take place. I mean, they're related to each other, right? Without light dependent reaction, there is no light independent reaction. So they both rely on each other. I mean, it's not like that if there's no light, then this reaction is gonna still take place. No, there's nothing like that. I mean, when there's gonna be light, this reaction is gonna start, and then when there's not gonna be light, this reaction is gonna continue. So this is a whole process, okay? They both depend on each other. Now, what happens in the light dependent stage? I'm gonna give you guys an overview here. So later on, we are also gonna learn in detail. So what happens in light dependent stage? Now, you guys can see that there is a photolysis taking place here. I mean, when light falls onto the thylakoid membrane, you guys can see here, right? This is the grana. I mean, this one is the grana. And in this grana, there are two photosystems, photosystem 1 and photosystem 2, which I told you guys. And what is happening here is when light is falling on the grana, the water is splitting up. Photolysis, photo is always means light, and lysis means breakdown. So photolysis is the breakdown of light, breakdown of water with the use of light. So water is broken down into hydrogen, oxygen, and electrons. When this happens, the hydrogen it reduces the NADP. NADP is another carrier that is present in the plants. It's a hydrogen acceptor. So what happens when hydrogen is released? It attaches to the NADP and the NADP is reduced. Now, another process that takes place here is photophosphorylation. Now, this process is really important. This is the main process that we're going to learn about. This is the main process that helps in photosynthesis. So, due to this photophosphorylation, which occurs in the photosystems, both of the photosystems, the ATP is produced. And this ATP and NADPH, which are produced in the light reactions, are then transferred into the light independent reactions. In the light independent reaction, a Kelvin cycle takes place. And due to this Kelvin cycle, we get our carbohydrates and the rest of the organic molecules, for example, like sucrose, cellulose, and a lot others. So, and a result of light independent reaction, what happens is we get our electron carriers back and ADP back and an inorganic phosphate. It's basically the most, it's like the opposite of the light dependent reaction. Also remember that in the light dependent reaction, H2O is a reactant and oxygen is always the waste product in the light dependent reaction. So yeah, now we're gonna learn about cyclic and non-cyclic uh, phosphorylation, photophosphorylation. I told you guys that there are two types, right? So the first one is gonna be cyclic photophosphorylation. Now, what happens in the cyclic photophosphorylation? You guys can see. Let's analyze the diagram first, okay? You guys can see there are two diagrams here. Photosystem. This is the photosystem one, the green one, and the gray one is photosystem two. So what is happening here? We're gonna go in order. No need to be afraid or be um, afraid from this, diagram. It's a really simple diagram if you learn it in order. So it starts from photosystem one. Cyclic photophosphorylation. Remember, always remember that cyclic photophosphorylation always occurs in photosystem one. It does not occur in photosystem 2. It does not involve photosystem 2. They're also going to ask you the difference between cyclic phosphorylation and non-cyclic photophosphorylation. So remember that cyclic always occurs in photosystem 1 and non-cyclic takes place in both of the photosystems. So yeah. So now what is happening in the cyclic photophosphorylation? Here you guys can see in the photosystem 1, light falls. When light falls, of course, the electrons that are present inside the photosystem 1, they get excited. Okay, the primary pigments, when they absorb light in the photosystem 1, uh, when they absorb light, the electrons present in the photosystem 1, they gain energy and they become excited. When they become excited, they are released, and this is a proton pump here. This proton pump also acts as an electron acceptor. So what happens is that these electrons which are excited, these are then accepted by these proton pumps. When these are accepted by these proton pumps, hydrogen moves from the stroma into the thylakoid lumen, right? You guys can see the down one is the thylakoid lumen and the stroma is the liquid, I mean the cytoplasm or the stroma, the liquid outside, right? And these hydrogen ions, these gray ones, are present in the stroma. When the electrons are, when the electrons become excited, they're accepted by these proton pumps, and due to the acceptance of these proton pumps, the movement of these hydrogen ions becomes easier, and it helps when the electrons are accepted, they release energy, and these hydrogen ions are then therefore transported into the thylakoid lumen. When the hydrogen ions are transported into the thylakoid lumen, these hydrogen ions then go to ATP synthase, and of course, I mean, when there's a high concentration in the thylakoid lumen, they're going to move outside. So when they're going to move outside down their concentration gradient, they're going to go into the ATP synthase. And as we learned in respiration, that ATP synthase, of course, it revolves and then it ATP synthase revolves, and then ADP molecule joins together with the inorganic phosphate to form ADP. And then these hydrogen ions are back into the stroma, and then from there they're pumped back into the thylica lumen. This process goes on and on. Now, when these electrons here, when they were accepted by these proton pumps, they went to these proton pumps, and then they came back into the photosystem 1. Now this process continues over and over again. I mean, photosystem 1 to the proton pump, then back to the photosystem 1. Now this is why this process is called cyclic, because this process repeats over and over again. Only for these electrons, remember, not for these hydrogen pumps. Okay, these hydrogen ions. These hydrogen ions, they go into the thylakoid lumen, then back into the stroma. These electrons, they always go from photosystem 1 into the proton pump, then back into the photosystem 1. So yeah. Now, you know that when these when these electrons are released, the hydrogen is also released with them. So what happens here is basically when these electrons are released and the hydrogen from the photosystem 2, when this hydrogen and when this electron, they combine together with the NADP molecule, which is a hydrogen acceptor, of course. When they combine with the NADP molecule, they form NADPH. And this is how this occurs, which is I'm going to explain to you guys that in the photosystem too. But yeah, for now, just learn that when these electrons later on in non-cyclic phosphorylation, when these electrons, the same electron, these combine with the hydrogen ions, and hydrogen ions that come from the photosystem too, which I'm going to tell you guys, they combine with them to form an NADPH. Okay. And also remember that this is chemiosmosis. When I told you guys that hydrogen ions are pumped back into the stroma and the ATP synthase rotates, so this is chemiosmosis. What was chemiosmosis? It was the process by which energy is supplied by the movement of hydrogen ions, right? Energy is supplied for the synthesis of ATP by the movement of hydrogen ions. It is not supplied from some other reaction. It is generated by itself. So yeah. Now we're going to learn about non-cyclic photophosphorylation. Now what happens in non-cyclic photophosphorylation? It involves basically both photosystem 1 and photosystem 2, as I told you guys before. Photosystem 1 comes before, which is PS618, yeah, normally. So yeah. In the step 2, what happens is that light is absorbed by both of the photosystems, and then this light is passed to the primary pigment of each photosystem. Now, I told you guys whenever light is absorbed by the accessory pigments, it is given to the primary pigment. So this is what they're telling, that that the light is absorbed and then it's given to the primary pigment. Now, after that, I'll show you guys that when the primary pigment absorbs this light, the electrons become excited, right? Just same as the cyclic photophosphorylation. The electrons become excited. When these electrons become excited, they're promoted to another energy level. And when this happens, in the step 4, what happens? When these electrons are excited, they go back into the proton pump. This proton pump you guys can see here, right? Wait. Yeah, this is the proton pump you guys can see here. These electrons, they become excited, go back into the proton pump. When they go back into the proton pump, they are then pumped into the photosystem 1, which is right here. Let me show you guys. Yeah, this is the photosystem 1, and this is the photosystem 2. So when these electrons, they're pumped into the proton acceptor, these electrons, then they are promoted into the photosystem 1. Okay. When they're promoted into the photosystem 1, they are then that process of cyclic photophosphorylation occurs. What happens is that the electrons are pumped back into this proton, then the hydrogen they're gone, then the hydrogen they're pumped from the stroma into this thylakoid membrane here, and then the thylakoid membrane is then, I mean, you know, the hydrogen ions, they go into the ATP synthase enzyme, and then that chemiosmosis occurs. So what happens here in in the non-cyclic photophosphorylation is that the electrons, the light falls onto this photosystem 2 first, and then photosystem 1. Due to this, on photosystem 2, remember that there's photolysis occurring here, so the splitting up of hydrogen will take place, and then these electrons will become excited. There will be promoted to energy level, right? I mean, they will be promoted to energy level after the energy is transferred to the primary pigment, right? So when it's transferred to the primary pigment and they're excited and they promote it to another energy level, this process is called photoactivation. Okay? Remember that this process of electrons being excited to a higher energy level is called photoactivation. After that, when these electrons are they're transported into the photosystem 1 from photosystem 2, um, when the photolysis occurs here, the electrons that are lost from the photosystem 2 are then replaced back by the photolysis. I mean, of course, when photolysis occurs, some hydrogen ions and electrons and oxygen is released. When these electrons are released, they replace the electrons that are lost from the photosystem 2. Okay, so in the last step, what happens in the photosystem 2 after the replacement of the electrons, the excited electrons from photosystem 1 and hydrogen ions from photolysis of water both combine with NADP to form reduced NADP. Now, you know that in photosystem 1, and I told you guys that in photosystem 1, the electrons are released, right? Like in the last diagram, let me show you guys here. In the last diagram, it was shown that these electrons are being released here into, these electrons are being released here, and there was an NADP molecule here, right? Which is hydrogen acceptor, of course. These electrons are being released, and the hydrogen is also being released from the photosystem 2 after the photolysis, right? So there's going to be hydrogen coming here. So now these three are going to combine, this hydrogen, this NADP, and electron. When these three are going to combine, NADPH is going to be formed. So this way NADPH is formed. I mean reduced NADP. So this is the final products. I told you guys that after chemiosmosis, ATP is formed, and after this process occurring here, after the release of electrons from photosystem 1 and the release of hydrogen from photolysis, there's gonna be this reaction is gonna take place when these hydrogens and these electrons are gonna combine with NADP electron carriers to form NADPH. Now these two are the products of what we call the light dependent reaction. Now these two are the products of light dependent reaction, and they go further into the light independent reaction as reactants. So that's all for this video, guys. I hope you guys enjoyed today, and um, I'm really glad to be releasing my third video. Thank you so much.